Next up, we have Andrew Lonsdale. Andrew is a PhD candidate using bioinformatics approaches to study plant cell walls in the ARC Center of Excellence in Plant Cell Walls. He is a software and data carpentry instructor and is also involved in community groups such as the Australian Bioinformatics and Computational Biology Society. Please welcome Andrew and his talk, Python for Science, Side Projects, and Stuff.
Thank you very much for the introduction, and thank you to the organisers for this chance to talk at PyCon. So I'm going to do Python for Science, Side Projects, and Stuff. And from the title, you might be expecting a bit of a mixed bag of a talk. You could be right. But what I want to do is I'm going to frame it as a story. And it's a story of a side project, how I got distracted from that side project, eventually returned to it, and in the middle, learned a whole lot of Python. But first, let's start with some definitions. So I'm saying it's Python for science. Now, I think science is more than just doing the science. And there's being a scientist and also just doing science. And I think we heard the mini-conf yesterday that Python is really good for science. But what I'm going to try and get across in this talk is that I think Python is also good for being a scientist. When I say being a scientist, I'm talking about things like sharing your data and teaching, networking in a social sense, communication, publications, and visualizing your results. So science, being a scientist, side projects, collaborations or projects outside the scope of your research, sometimes very, very far out the scope of your research. And also stuff from other Python packages I've used along the way in passing, or at least I plan to. And side projects are a tricky thing. Sometimes you have to concentrate on your research, and sometimes there's a little voice that says maybe you should just explore this idea and see where it goes. And I like to think that that voice comes from the side project devil. So when you listen to side project devil, you go off and you try other things. And some of those other things I tried have led to me learning a bit of Python. So I said we're looking at all these things. The first thing I'll talk about is teaching. So I think Python is great for teaching. I said I'm a software carpentry and data carpentry instructor, and I also teach with Combine, which is the student and early career researchers group for computational biology. And as you may have seen associated with PyCon, there was the pre-PyCon data carpentry we had a couple of days ago. And it was a really good experience, and a lot of people from PyCon and also others interested in Python. It was a 2-day course looking at data carpentry and using Python primarily to teach the skills of data cleaning and data analysis. Give a shout out to Alistair and Daria and Sam, who were the main drivers of that, and a few of others who helped teach. But it's a really, really good opportunity. And with teaching in Python, I think the trick is that Python is a great language for learning, which means there's lots of people that want to learn it. And so if you know it, you get an opportunity to teach. And if you're in an academic field where there's not many teaching opportunities, it could be a small field, or it could be an emerging field, then it gives you a chance to teach that you might not otherwise get. And there's always constant demands for these kinds of workshops. The other great thing about being a scientist that Python can help with is communication. Presentations like this one are a key part of academia. You have to present your research to get feedback and also promote the work that you're doing. I'm always tempted to use fancy things like Reveal and Rise live demos of Jupyter notebooks and things like that to have a reproducible presentation. But in the end, it's hard to use these in an existing environment where people expect PowerPoint files. They want to make comments on them, send them back, and I've had at least one live demo go wrong with trying to use a live notebook. Everyone expects PowerPoint files, and so you might say, look, just go manually and use PowerPoint as one solution. The other one is Python, is to use Python and go install Python pptx. And you can do that like I did for the first 5 slides of this talk, and you can see that I actually made them all just using pure Python. So you can set your title and your subtitles and add some images there. And when you're sharing your results, I think that's a great way of, if you're doing all your analysis remotely, you can collate them all, package it into a presentation that might be in a nice format for supervisors or collaborators and send it off without sending it in a PDF or saying it's on the web, you can download, and then your supervisor says, how can I comment on that? And then you have a long conversation. So I think that's really useful. But I suppose I'll digress now and say, well, how did I actually get here? Why am I talking about being a scientist and science? And I actually came to bioinformatics a roundabout way. So I originally studied software engineering. I was working in web hosting and domain names and decided I wanted to change in my life. So I had a big career change a few years ago, and I went back to study bioinformatics, and then I continued to work and study in bioinformatics looking at plants. Now, a few of you might have seen a talk yesterday by Daria on bioinformatics, and I think there's another one going on now. And there have been a lot of good official definitions about what bioinformatics is in this conference, but I thought I'd try and get it across succinctly. So for me, bioinformatics and working in plants is essentially this. What you're doing is you're getting information about organisms. In this case, I'm getting information about Arabidopsis, which is a well-studied organism in the plant world. And we use Pokeballs or sequencing machines and mass spectrometry to get information about these organisms and then study them. And my research in the Centre of Excellence in Plant Cell Walls is all about using computational approaches to look at plant proteins. And computational approaches for me really didn't always include Python. So in my old life, I did mostly Perl, a bit of PhD, and I did a little bit of C and Java in my studies. Now I use Python pretty much for everything. I'd never really used it much before, so the question is, what happened to me? And I'll try and frame the rest of this talk as a bit of a tale of two papers. So there's a paper published in 2014 and earlier this year, and I talk about how there was a whole lot of Python all in the middle, and how this sort of opened the floodgates for me using Python in my research and around my research, and some of the things I've learned along the way. So it started really with a journal club. So we meet at the unsuitable time of 8 a.m. every week, but for some of us that's the only time in a week we're on campus at 8 a.m, so it helps. And one week in this journal club a few years ago, we were looking at a collection of papers called the 10 Simple Rules collection. This is published by PLOS Computational Biology, and it says there it's a quick, concentrated guide for mastering some of the professional challenges research scientists face. We read a few of the articles, we thought they were quite good, and we were pretty pleased with our journal club, how it had been going so far, so we thought maybe we could contribute. We started a pretty rough draft of how our journal club had been going, we collected some data, and then Harriet said, this actually would be a lot easier if there was 10 simple rules for writing one of these 10 simple rules. That comment quickly escalated. So later that year, this came out, and it was a lot of fun to write. It was my first publication. It's not what I expected my first publication to be, not what my supervisors expected my first publication to be, but it was a lot of fun. And apart from the writing, it was a fairly casual tone, had a lot of fun with the plots in it. And like good researchers, we tested our assumptions. We said, how many rules are in a 10 simple rules? 10. Now, if you might recognise that plot, it's pretty much adapted straight from the matplotlib examples gallery, and that, of course, is based off my classic XKCD on there. But along the way, when I was testing this silly question of how many rules in a 10 simple rules, I learned some things. So I used requests and beautiful soup in Python, and I was pulling down the author information on each of these articles, counting how many rules are in them, taking out the authors manually, and creating a few more plots for the publication. So we looked at the authors, and we looked at the founding editor of the series, and we found that it definitely helped to be him to get an article in that series. When he decided to collaborate with us, that was really good because it put us in this section rather than the also rans. Another thing we did is we looked at the impact of these articles, and you can see that nobody cites them at all. but everybody reads them. And I think that's the point, that it's a very casual and gentle introduction to science and doing science, and I think that's why it's a really popular series. So I learned a few things from that experience, that SciPy project, and as one was the power of Python. I delved into the API back for this plot to actually pull in the amount of information, citations, and how many views via the article level metrics that PLOS at the time had via their API. It's slightly changed now, but it's still out there. And of course, I learned to use plot.xkcd. And really, this was the start. And matplotlib was my gateway package. Once I started using that, I was hooked on Python, and it's just gone on from there. After that, I did some Venn diagrams for some of my research. I thought I'll try Python because I've just used it recently, and it worked pretty well. Import matplotlib Venn. And then I thought, well, if it's matplotlib Venn and it's using matplotlib, I wonder if you could take another XKCD comic and if you could make that as well. And the answer is you can. So just by importing matplotlib and with a little bit of hacking to work out exactly where you want the bits of text to appear, you can relatively easily recreate that comic in Python. It's a trick I still use today, and so on those first 5 slides, they weren't images, they were plots as well. And again, it's not very hard. The hardest bit is getting the font download, and there's a little hack there to use it locally. But otherwise, you can create any Venn diagram you want, use plot.annotate to put little bits of information on there, and it's quite easy. And Venn diagrams are pretty common in science, and so it is a trick that I think I'll use more and more. As said in the 10 super rules, we looked at the metrics of some of those articles, and there's a few other APIs around there. One day I thought, instead of going to Google Scholar all the time, I wonder if it'd be easier to pull information down. But one of the lessons I've learned is in Python, someone's almost already done it. And so if you look, scholar.py is out there. It's a gentle web scraper because Google obviously don't like or want or allow via their terms of service people to download all of Google Scholar. But you can do fairly casually, if you just put in questions about an article or who cited it, you can pull the information out of Google Scholar automatically with that. Another thing about being a scientist that I think is really interesting is communicating not only with your own group, with others, but within your own group. And so we've come across Slack, and many research groups are starting using Slack. And of course, there are Python libraries for interacting with Slack. And so we've started a few little Slack bots, and they're online there. So one interacts with the Scholar script from before, so you can build a Slack bot that says, who cited our great paper, 10 simple rules for plus 10 simple rules? And a Scholar bot will give you back the link straight away of all they want. So that's online. If anyone wants to can think of any more, please feel free to add to them. One more thing about being a part of a science is networking socially and things like conferences. And at conferences, you can have a great talk, like this one, or you can have a bad talk, possibly like this one, and you might be on Twitter and you're going away. But even if you're on Twitter right now or on the PyCon channel on Slack, you could be interacting with each other. You could be collecting data about what's going on Twitter or Slack, and you could interact with people. So I've done that at conferences, and you can see who's tweeting the most. And then, of course, you can fall back on your crutch of matplotlib and plot who had the most tweets, and that was through a genomics technology conference a couple of years ago. After learning all of these things, finally, it was time to get back to the thing that we originally were going to do, which was write the article about 10 simple rules for a journal club. And it took 2 years later, but in the meantime, I learned a lot of Python, and I said we kept the data, and so I went to my old trick, of course, of plotting how many people had turned up, making sure that everyone knew that reading alone was not really a journal club. The question, of course, that you and supervisors might ask is, has it actually helped? And I firmly believe it has. So playing around with matplotlib, I think a lot of research visualization I do now, I've got such fine control. If I'm doing custom plots, I know exactly that I can change the color or the shading of any point on the plot, and I've got a lot of confidence in how to do it. The web scraping and form submission skills that I learnt interacting with the 10 simple rules lists of articles have come in really handy looking at existing tools that are out there. So many science tools are still on the web. It's a single server. It was written in PHP or Perl 10 years ago. No one's going to get any credit to update it, and it's going to stay online as long as the server's online. So if you want to get results from that server, you have to either manually post in single sequence after single sequence, or you need to write a script. And so using things like Robo Browser and requests, you can take a whole lot of information that you need to get results on and send them one by off overnight and not have to worry about it. I said API calls was a great introduction because there's lots of public data out there. And so although there are examples of these older API-less services, there's a lot of data out there that's got really modern APIs. So you want a language that people have built tools and libraries for to use those kinds of things like RESTful APIs. And of course, I think just my general programming in Python in general has improved. So things like decorators, I hadn't really come across them before, but the Slackbot library uses it extensively to work out how to reply to individual threads and things like that. So I thought of 10 simple rules for side projects as I was writing this talk. And the 5th rule is you can start it anywhere. It doesn't have to be at the start. It's just a side project. Rule 2 is you collaborate, because if you do it on your own, sometimes you forget. And I found a lot of half-finished things preparing for this talk, which I finished for this talk. And rule 9 is you don't have to finish. And you can come back later on in any order that you want. So in summary, I think Python is really good for scientific research because it's the other things that you can do around research that really help. You do what comes easy. So if you practice more with Python, the next time you come up with an obstacle, you're not going to go to Excel because say, I'm only going to do this once. If you're really comfortable with Python, you'll say, I'll do it in Python because it's easiest to do in Python. And then you'll always use it more than once because that never always happens. And I said, you never actually know what skills you're going to need. So you may as well get all the skills and hope that you'll use them later on. So a little bit of logic at the end. Side projects are good for Python. Python is good for science. Python is good for being a scientist. So therefore, side projects are good for science and being a scientist. It's infallible. I'd like to really thank one person in particular, Harriet Dashnow. So she was co-author on both papers and helped a lot of the side projects. And a plug for her book, Elegant SciPy, is there. Just read it there. They obviously didn't invite me to be an author because Inelegant SciPy is not a great title. I'd like to thank everyone in the Journal Club that helped me out and Combine, the group there. I have one more thing, but it's a 0th thing because indexing at base 0. There are a lot of really good talks on at PyCon right now, including right now. So I think to the left we've got Python for bioinformatics, which I would have liked to have seen, and the power and responsibility of Unicode adoption, which looks really interesting. And everyone's here. So some of you, I wish I could go to both. Some of you may be regretting not going to one of those talks right now. So I was thinking, how can I help everybody out that didn't get to go to those talks? And the solution, of course, is a side project. So FastQ is a format in bioinformatics, and emojis are emojis. So we represent biological sequences as letters in bioinformatics, and it's based on the first letter of the chemical name, ACTG, typically for DNA. That makes sense, but it's pretty abstract. So why couldn't you use emoji? And the answer is you can. So in Python, you can use PyEmojify and import emoji. And so A's, we'll call them apples, C, corn, T, tomato, G for grapes, and if we don't know, we'll put a question mark. We've got 2 typical sequences here. We write a little function that we just map everyone back to its emoji, join it, and print it. And so these 2 sequences here, we can represent them as emojis. I should have stopped there. But then I thought, if I'm making something, it actually should be useful. So FastQ is a file format that has faster. So faster just has the letters, ACTGs. But the FastQ format also has quality information embedded inside it. And you can think of that as the sequencing machine, or the Pokeball from the slide earlier, how confident it is that each base is the correct call. And so you generally have these, I think about 93 scores, increasing from low quality to high quality. And that lets you know how likely it is that the sequence you've got there you should trust. And when you're taking these individual reads and aligning them back to the genome of an organism, you can use the number of high quality reads to make good calls, and you can discard bad calls based on the individual qualities. And often you'll take these files and you'll use a program that will give you lots of plots and tell you the average quality of all the sequences you've got, and that's a good way of doing quality assurance. But I thought it'd be good if you could do that on the command line, and if you could do that with emojis. And so what you do is you take every single bit of quality, and so use skull and poop and no entry sign for the bad ones, and smiley faces and sunglasses and grinning for the good ones. And then you do the same thing. So take these two pairs of sequence reads, and we're taking the mean of every single position. We can see that overall we've got some quality issues at the start, which is actually typical of the machines, and also got the quality drops off towards the end of the reads as we sequence them. And again, that's typical. So in one line, you can quickly look at the quality of all your data and then run some trimming to cut off these things. So that used some sample data from the VLSCI's online tutorial, and that's online if anyone wants to make it even better. I was thinking you could do all summary statistics and do mean, median, and it would be great. So if anyone wants to help, that'd be great. And I think that's the last slide, so thank you.
We've got some time for questions if anyone has some. No questions? That's a perfect talk.
That's the interpretation I'll take, yes.
Yeah. Really? Okay. Well. Oh, yes.
What's your next project? My PhD. That's my supervisors who are hopefully not watching this online.
You're welcome to ask him questions after as well. Could we please give him another round of applause?